I'd like to thank the organizers for allowing us to present our work here. I'd like to say at the outset that I'm presenting for Dr. Latha Satish, who unfortunately couldn't be here to present this work, so I'm sort of the pinch hitter. I have nothing to disclose, so I guess I'll leave the podium now, but not actually, but nothing financial anyway. And we're going to have a change of pace because my talk is actually to talk about trying to establish an animal model for Dupuytren's. One of the things that we feel has sort of been an impediment to the development of new therapies of Dupuytren's is the lack of a very good animal model in which these things may be tested. And so we decided to try and see if we could create one. There have been attempts at animal models previously, and I guess we'll be hearing some more about this in subsequent talks. Initially, in the 60s, in monkeys, there was an attempt with disruption of palmar fascial fibers. Then Dupuytren's contracture tissue was placed into pockets in the suprascapular areas of nude mouse in the 80s. And more recently, a similar scheme was used in nude rats and tested. So the aim of our study was also similar, and we wanted to establish an animal model that may ultimately serve as a platform in which to compare treatments for Dupuytren's contracture. Now, if we hypothesize, as we've heard here, that fibroblasts/myofibroblasts are the cellular effectors of the Dupuytren's phenotype, what we sought to do was then to take the cellular effectors of the Dupuytren's phenotype, not just the tissues, but the actual cultured cells from Dupuytren's patients, and put them into the forepaws of nude rats to see what their behavior would be over time, and whether we could recapitulate a Dupuytren's type of phenotype. Now, we knew that Dupuytren's was an insidious disease that required quite some time to manifest, and we expected that that may be the case in an animal model as well. And we didn't want to just put cells into the animal models and then sort of wait blindly, wondering whether they were there, whether they were behaving, whether they were staying localized, etc. So we decided to try and trace them. And to trace them, we first introduced a lipophilic fluorescent cell tracer into the fibroblasts in vitro. And what you can see here is that here are cells that have been transfected, not transfected, it's really just a DMSO treatment with this molecule to introduce it into the cell membranes. Here are cells that were not, and when you look at them with a charge-coupled camera device, then you can see the fluorescence that has been created by that cell population. So these were our first attempts, and what we did was take nude rats and inject cell populations from fibroblast populations from Dupuytren's patients into the forepaws of the nude rats in a subcutaneous plane. And then we followed them over time, because what we could see was that, yes, we could see the fluorescent localization of that cell population, and that it stayed confined to that anatomic location. And we did see a decrease in signal over time, generally speaking, and we followed these rats out for somewhere around 8 to 9 weeks in general. Why 8 to 9 weeks? Well, we needed to establish an endpoint for experimental practicality, but we wanted to give it some time to allow any sort of changes to accumulate and to give us the opportunity so that when we did subsequent analyses, we would be assured that the results were reflecting the physiology of the cells in the transplanted environment. What you can see here in these initial results is that we assayed after the animals had been allowed to persist for roughly 8 weeks. We harvested the subcutaneous tissues, we harvested the skin of the forepaws separately. And what you can see here, when we looked by RT-PCR for human alpha-smooth muscle actin, so these are probes for human alpha-smooth muscle actin, at 2 different cell concentrations, we had very high levels of alpha-smooth muscle actin expressed, human type 1 collagen expressed. In the rat's skin itself, there was none, and when we just used control agarose medium, then there was none as well. So the next step for our model, having convinced ourselves that cells would persist and that they would, in fact, still look like myofibroblasts at the end of that period of time, was to compare fibroblasts taken from carpal tunnel patients with fibroblasts taken from Dupuytren's patients. So we had a series of animals in which in one forepaw we injected 1 × 10⁷ cells of carpal tunnel fibroblasts, and in the other we injected 1 × 10⁷ cells of Dupuytren's-derived fibroblasts. And in both populations we were able to follow the fluorescence signal over time. And once again, we did see that there was some decrease over time, but it was still quite easy to identify even at the end of 8 to 9 weeks of animal persistence. Here's another animal showing again that the fluorescence signal can persist. Here's a third showing again the persistence of the fluorescence signals. And then at the end of that period of time, we harvested again skin and subcutaneous tissues separately and started to subject them to some histological and molecular analyses. The first thing we did was to look at the histology, and what I'm showing you here is that in the top panel you see the results of H&E staining and Masson's trichrome staining from the subcutaneous tissues of carpal tunnel injected forepaws, and here are the results from the Dupuytren's injected forepaws. I think it's quite clear that there is substantially increased cellularity in the Dupuytren's-derived forepaws compared to the carpal tunnel-derived forepaws. So that was one striking finding. The Masson's trichrome, again, showed that there was substantially denser, as you can see here by the black and by the increased thickness of collagen that presents, there was substantially denser collagen in the Dupuytren's-derived forepaws than in the carpal tunnel-derived forepaws. Now, we were also interested to examine these for molecular markers that may be associated with the formation of Dupuytren's disease or a contracture phenotype. And of course, the first one that we were interested to see was alpha-smooth muscle actin. So what you can see here is results of quantitative RT-PCR reactions on the populations of Dupuytren's-derived forepaws versus carpal tunnel. Here are a similar number of carpal tunnel and Dupuytren's-derived cells in culture. So in culture, the Dupuytren's-derived cells seem to express some more alpha-smooth muscle actin. The skin control, again, showed none. No human-derived alpha-smooth muscle actin. The carpal tunnel fibroblast actually had very little alpha-smooth muscle actin at the conclusion of these experiments, but the Dupuytren's-derived fibroblasts, after some 2 months of transplantation into the forepaw, had very high levels of alpha-smooth muscle actin. In contrast, when you looked at fibronectin, that was not the case. Although in culture, the carpal tunnel and the Dupuytren's-derived fibroblast appeared to have differential regulation of fibronectin, again, nothing was seen in the skin. In the forepaws, there was no significant difference in expression in the amount of fibronectin messenger RNA. We looked also at type 1 collagen mRNA expression. Once again, here are carpal tunnel and Dupuytren's-derived fibroblasts in culture. And both carpal tunnel and Dupuytren's-derived cells in the forepaws demonstrated increased type 1 collagen mRNA compared to their expression in culture, but the Dupuytren's-derived fibroblasts had a vastly increased expression of type 1 collagen in the forepaws. But that was not true of type 3 collagen, which we looked at as a control. Here in culture, the Dupuytren cells and the carpal tunnel cells seem to be similar, and after transplantation into the forepaws, these cells also appear to be quite similar in the levels of their expression. So I will summarize these initial observations for you by just pointing out that, number one, human-derived fibroblasts will persist for lengthy periods of time in the forepaws of nude rats, that we can, at least with a little bit of confidence, use in vivo imaging to reassure ourselves of the cell fate of these transplanted fibroblasts in an animal model for up to 8 weeks. We again saw increased cellularity in the forepaws, apparently increased cellularity in the forepaws receiving Dupuytren's-derived fibroblasts. And Masson's trichrome stain showed increased collagen staining. The alpha-smooth muscle actin type 1 collagen was increased, and fibronectin and type 3 collagen was not increased in Dupuytren's versus carpal tunnel-derived cells. We did not, at the end of this period of time, see an actual contracture. It would have been great if we could have actually seen some kind of grossly visible contracture forming in these animals. It is possible that if we let this process go on for longer, that one may develop. And we are in the process of sort of observing a cohort of animals to see that. We're also expanding the sets of genes that we're looking at in these situations, but we're hopeful that with further examination that this kind of a model system can be an animal model in which various treatments for the actual physiological / molecular mechanisms that are driving Dupuytren's can be evaluated. I want to thank the multiple collaborators that we've had on this and other Dupuytren's research, some of whom are in the room today, as well as some of our funding sources. And I especially want to, once again, bring to your attention Dr. Latha Satish, who should have been here to present this work and will undoubtedly join us all for the next Dupuytren Symposium. Thank you for your attention.